We now move to North Carolina, and we have a paper on Staphylococcus aureus, which will be presented by Dr. Kevin Anderson. Staphylococcus aureus in dairy goats and human milkers in North Carolina. Kevin.
Good morning. Good morning. Get oriented. Okay, start out why study Staph aureus. My lab and myself have been interested in Staph aureus in dairy cows for quite some time. And in North Carolina, we have a considerable number of problems with Staph aureus, and particularly in heifers and particularly in some herds. Our interest in dairy goats is that Staph aureus is a major pathogen in dairy goat mastitis infections. It can cause a severe gangrenous mastitis, and you see an example in the slide here in a ewe, and you see the lower purple area, which would be cold to the touch, and I'm sure many of you have seen gangrenous mastitis from Staph aureus. Although that's dramatic, the most common manifestation of Staph aureus is subclinical infections that can be contagious and persistent. Staph aureus can also be a zoonotic pathogen, causing skin and other infections in humans, and it can cause food poisoning. One of the things that makes it difficult to deal with is that there's multiple sources of infection, and it's easily transmitted. It's found on body sites other than in the mammary gland, and it's found in numerous mucous membranes. We in particular have found it in North Carolina being transmitted by horn flies, blood-sucking horn flies, and we found the same genotypes of Staph aureus in horn flies as we find in mastitis infections in both heifers and cows in the herd. It can occur in the environment, and it can be spread through fomites such as water troughs and other areas where there's contact with secretions from the mouth, etc. Also, there's a potential for the spread and for the involvement of methicillin-resistant Staph aureus via transfer of the MECA gene. For this study, we visited 30 dairy goat herds in North Carolina in fall of 2014 through July of 2015 and collected 519 goat milk samples. North Carolina largely has small backyard operations. The largest dairy goat operation has about 1,200 milking does, but most of the operations are backyard. The 519 samples were usually composite samples of the two halves of the udder, although if we had a suspected mastitis problem, we would collect both halves. We did not exclude animals that were showing signs or that the owners reported had clinical mastitis at the time. We also collected swab samples from the nose or nares of the goats, and then we collected hand samples and nasal swab samples from the human milkers that were available. And we collected some information from the producers on management practices and herd demographics. As I said, there's a lot of small backyard operations, and the bulk of the samples were collected from these small backyard operations. Only milking does were sampled, and we did not collect more than 100 does from a herd. Herd size ranged from 2, large herd, right, to 235. The median was 8.5. Predominant breeds were Alpine and Nubian, and the others are listed, but those are the most common in the U.S. And 90% of the operations had access to pasture as opposed to being confinement-based. In terms of biosecurity, all farms had pets and or other livestock on their farm. And there had been a lot of exposure to outside goats. In the last year, 2/3 of them had exposure to goats on other farms, either from going to a show or an exhibit, or from adding new animals, or from sending animals to other farms for breeding. The goat milk was used for a variety of purposes, and here there could be more than one use. 93% of the producers reported home consumption, 77% used it to feed animals, 60% made food products, the majority cheese, some yogurt and gelato, some non-food products, mainly soaps, and then 30% reported that milk was sold as fluid milk. And even though that's not legal in North Carolina, they reported it that way. It's called pet food sales. They sell it and it says pet food. 93%, all but 2 of the individuals reported that they consume raw goat milk, so very frequent. Okay, the lab techniques, we plated 1/10 of a ml of milk. The standard inoculum is more like 1/100 of a ml. This was a larger volume. It allows detection of lower numbers of organisms, so a lower detection limit. We used procedures that were consistent with National Mastitis Council recommendations. The swabs were macerated in saline, and then we used Staph Express Petri film plates. And these were validated to be highly successful in recovering Staph aureus as compared to reference techniques. Susceptibilities were done, but they were just Kirby-Bauer susceptibilities. We genotyped the isolates using pulse field gel electrophoresis after digestion by SMAW1. We analyzed the gels and made dendrograms using bionumeric software, and the statistical analysis we used the Wilcoxon signed rank test with SAS statistical software. Milking procedures, the majority milked 2 times a day by hand, machine, or a combination. There was a rare individual that milked 3 times a day, and then some that only milked once a day. 70% of the producers never wear gloves. Most used a post-milking germicide, contents given there, and then over half used a pre-milking germicide. The majority reported using antibiotics for treatment of clinical mastitis, but only as needed, and only a little over a quarter used intermammary antibiotics at dry off, and that's pretty consistent with what we find in that area. 30% of the milkers worked in health-related fields, and later when we studied that, we did not find any association with the presence of Staph aureus or the resistance of the isolates. Okay, the most surprising result, and this goes along with the story that if you want to make sure you can't find something, you should study it, is we found very little Staph aureus positive goat milk. Only 1.2% of the milks we tested were positive, so very little Staph aureus. Those were not very resistant, and in all cases, when I talk about resistance, I'm talking just to 1 or more antibiotics. So only 28% were resistant to one or more antibiotics. Surprisingly and consistent with other studies, we found that goat nasal swabs from these same goats were positive in a considerable portion of cases. 42% were positive, about the same level of resistance. A fourth of the human hand and nasal swabs were positive, and they were much more resistant than were the goat swabs. And the specifics of that is given. Our 1.2% positivity rate is really lower than what's reported in the literature in terms of Staph aureus from dairy goat operations. Looking at some of the associations, we found that larger herds had a greater proportion of nasal swabs that were Staph aureus positive. Machine milking was associated with a greater proportion of Staph aureus positive milks, but the reliability of that is questionable because of the small number of positive samples we had. Herds using pre-milking germicides were found to have a higher proportion of resistant goat Staph aureus positive nasal swabs. Not sure really of the significance of that. Whoops. Okay. I'm not going to spend a lot of time on this, but this is a dendrogram of the results, and basically, make sure, okay. The top is the more resistant isolates. These are the antibiotics, and if there's a gray area, then that indicates resistance. The lower ones are primarily the goat isolates with hardly any resistance, and they're very monotonous. These are the bands after digestion, and they all are nearly the same exact genotype. So the resistance we found was largely in the human isolates, and they were genetically distinct from the animal isolates. And he's telling me my time is short here, so I'll pass that and just go to the conclusions and say consumption of raw milk in North Carolina is very common. Low Staph aureus presence in milk. Is this due to the successful application of management barriers to infection, or is it the different behavior of the Staph aureus genotypes that are associated with goat mastitis? It was interesting that there was so commonly that goat nasal swabs were positive, yet very little in the milk. Okay, so obviously it's a different epidemiology associated with that. Resistance patterns primarily were associated with the human adapted or the human strains of Staph aureus, not the goat adapted Staph aureus strains. And it suggests the real potential for transfer of resistance is from the humans to the goats rather than from the goats to the humans. One very interesting thing is similar to what we find in cattle is coagulase negative staph were very commonly isolated from the goat milks. 34% of the samples were positive for coagulase negative staph, and they were fairly resistant, and that's also similar to the cow situation. And the species, just if you're interested, are listed here in decreasing order appearance, and then the number resistant over the number susceptible are given here as a fraction. So for a large proportion of the isolates, they were fairly resistant, meaning they were resistant to one or more antibiotics. and want to acknowledge the support of the American Dairy Goat Association Research Foundation and the goat producers and their North Carolina Dairy Goat Association, and then the support of the American Association of Small Ruminant Practitioners. And I would answer any questions. The picture on the right is Rachel Kearns, who is the veterinary student that collected a lot of this data. Thank you for your attention.
Thank you, Kevin. Take one question. I'm sure there will be. It's an interesting study. Certainly the nasal swab was, that data was quite interested in that, I think.
Okay?
One question, please, and just say where you're from. And the other thing, can I remind people to put their cell phones, mobile phones on silent? Thank you.
Hello, Mário Balaro, Brazil again. Did you find some correlation between people with resistance, bacterial resistance, and consumption of raw milk with some antibiotic results? Did you look for something like that?
We didn't specifically look at that, mainly because most of the farms reported that they consumed raw milk, but we didn't ask who. So you're asking if there's a specific association, whether an individual that had resistant Staph aureus also consumed raw milk? Is that what you're asking?
Yeah, because they could be using antibiotics in this goat, and these antibiotics going to the milk, and after that they are consuming this milk with residues. No?
Yeah. We did not ask that specific information. We didn't have approval to ask that much detailed information about the producers. So I'm sorry, I can't answer that.
Thank you.
Okay?
Thank you once again, Kevin.
Thank you.